Hi everyone, welcome to my channel Pure Biology, and I'm sorry for not giving any video for a long time. So for that I'm very sorry. Okay, let's start. Today's topic is the photomorphogenesis. First of all, not we learn the photomorphogenesis, we just learn which factor that helps in the photomorphogenesis of the plants. This is the obviously topic related from the plant physiology and important obviously for NEET exam. Next, I have to say that this is the photomorphogenesis is actually nothing but the growth and the development of the plant. Okay, we all know that the photosynthesis is the process that is done by the plants, but also, and this is obviously light dependent, and another thing, that is the growth and the development of the plant's body, that is also light dependent. And here you all know that the red or blue light that are very important for the growth of the plant's body. Okay, so here this is the the factor today we learn, that is the cryptochrome. Okay, cryptochrome. This cryptochrome actually is a chromoprotein. Okay, and it is help in the plant's growth and development. Obviously, I said that it is a photoreceptor. It is a blue photoreceptor. I told you that generally red and blue light that are very important for the plant's different activity. And here this cryptochrome, it is related to the blue light photoreceptor. Okay, blue light photoreceptor. And this is actually the two main subunits, or you can say the two important factors are there, that is pterin and flavin. Okay, pterin and flavin, that are very important factor and present in the chromophore. What is the chromophore? This is the cryptochrome, and the cryptochrome actually it has an important factor. This is called the chromophore. And what are very important for controlling or for the activity of the chromophore? This is the pterin and the flavin. Okay, and we find that generally three common flavins are there. Three common flavins. One is the riboflavin, and another two are the derivatives of the riboflavin. Okay, riboflavin main important factor, and another two important derivatives, that are FMN and FAD. Both are present. Next, this flavin, there is another protein present, the flavin. Okay, and this protein is called the flavoprotein. Okay, this is called the flavoprotein, and these are actually three important subunits, or you can say three important factors we learn here. What? First of all, this is the cryptochrome, is the chromoprotein. Okay, this is the chromoprotein, and obviously is a blue light photoreceptor. It is a chromoprotein and it is blue light photoreceptor. And this and the chromophore. Sorry, one second. Sorry for the disturbance. Okay. So what we learned here, that is the cryptochrome. It is the chromoprotein, and it is obviously blue light photoreceptor. That means its activity is totally dependent on the blue light. Okay, blue light, and it has generally one factor that is called the chromophore, that contains two another factor. This is called the pterin and the flavin. Okay, and in case of flavin, three types of flavins we can see. One main important flavin is the riboflavin, and another two are the FMN and FAD. These are the derivatives of the flavin. Okay, and there is the flavin that contains one important protein. This is called the flavoprotein. This is the general structure. Its proper structure we will learn here. Okay. So where actually these are found? These are found generally in all the plant species. Okay, in all the plant species, not the plant species. Obviously, sometimes it is found in the animals also. Okay, generally its activity in case of animals, it helps in the circadian rhythm. It helps in to control the or to complete the circadian rhythm. It is another term, and I will discuss about this circadian rhythm in my another video. Okay, this is another term. So just you know that you all just know it is what is the role of this cryptochrome. It is important for the animals for completion of the circadian rhythm. Okay. Next, actually these are structurally related to photolyse. What is the photolyse? Photolyse is one kind of enzyme that helps in the repairing of DNA. Okay, pyrimidine of, you know, all DNA contains purine and pyrimidine. So pyrimidine repairing, the main role of the photolyse is pyrimidine repairing. Okay. So cryptochromes do not have DNA repair activity. Okay. So what is saying here? This is actually said that the cryptochrome, the structural formula of the cryptochrome, that is just like the photolyse. And what is the photolyse? Photolyse is one kind of enzyme, you can say, and this enzyme actually helps in the repairing system of the DNA during what kind of repairing system? It's a help in generally pyrimidine repairing system of the DNA. Okay. So this is, but we don't see any repairing activity of the cryptochrome in the plant's body, obviously, or plant's DNA. This is the structural situation, or you can say the structurally same. You can say the structurally same. That means the cryptochrome is structurally same just like the photolyse, but its activity that is present in the photolyse, it is not seen in the cryptochrome. Okay, that is the DNA repairing system. It is not seen here. Next we learn here the structure. Before going to the structure, first we have to draw a rough sketch of the cryptochrome, and then we can understand it properly. Okay. So this is the structural rough sketch, you can say, of the cryptochrome. What is said here in the structure? Generally, cryptochromes, it has two domains. Okay, two domains. One is the amino terminal photolyse region, homology region. That is a short form of this PHR. And another domain is the carboxyl terminal domain. Carboxyl terminal domain. So what are the two domains present in the cryptochrome? Again, I am saying that this is the amino terminal photolyse homology region. The short form is the PHR. And second one, the carboxyl terminal domain. Clear? This PHR, this possesses two chromophores. In this PHR, the two chromophores are present. I explained before that or said that the flavin and the taurine. Okay, the flavin generally is the FAD, and its absorbance light is 450 nanometer. Clear? And the taurine is actually methenyl tetrahydrofolate. Okay, methenyl 5-10 methenyl tetrahydrofolate, and its short form is the MTHF. You can see here, this is MTHF, this is MTHF, and this is FAD, and this is FAD. Clear? So, and it can absorb generally 380 nanometer of wavelength, light wavelength. Clear? So these are the two domains, and between the two domains, only the first one, the PHR, it consists of FAD and MTHF. Clear? So we can say the cryptochrome is the dimeric protein. It is called the dimeric protein. Now we have to learn how it becomes active. The demelanization it is very important or essential for their activity, biological activity. Before going to it in active, demineralization is very important. Clear? Demineralization is very important, and that is totally mediated by light. And which domain is involved here? The sensory domain, that is the N-terminal domain. This is the N-terminal domain, and this is very important or sensitive to the light. Okay. So, demineralization is very important for the activity of the cryptochrome, and this demineralization generally starts in the N-terminal domain of this pigment. Clear? And in case of the C-terminal domain, that here is another subunit or factor, you can say. This is called the dash, and what is the full form of the dash is DQXVP acidic stage. Okay, DQXVP acidic stage. This transduces the light signal by interacting with the E3 ubiquitin ligase and COP1. Sorry, what is saying that its activity is the demineralization. Okay, demineralization, that means the light sensitive region is the N-terminal domain, and it is totally dependent on the light. But in case of C-terminal, what happens there? The signal, this factor is very important because this actually transduces the light signal. It transduces the light signal. How transduces the light signal? When interaction occurring with the E3 ubiquitin ligase and the COP1. What is called the COP1? The full form of the COP1 is what? This is called the constitutive photomorphogenic 1. Okay, photomorphogenic 1. So this situation is totally completed, then the cryptochrome become activated, and after that, and when this is possible, the two important wavelength is very important. This is the 380 nanometer and another is the 480 nanometer. The two subunit, or you can say two factors, flagging and the tailing, both are very important for absorbing this 380 nanometer and 450 nanometer of the blue light of sunlight. Blue color light from the sunlight. Okay, so this is occurring, total situation or mechanism is occurring within this pigment, and after that this cryptochrome become activate. Okay, and after going its full activation, the growth and the development of the plant's body is controlled. And two kinds of cryptochrome, so we can find generally CRY1 and CRY2, and the main difference between the CRY1 and CRY2 is that the CRY1 is stable and the CRY2, this is easily degraded or rapidly degraded in the light. Okay, so now we have to learn the actual function that is totally done by the cryptochrome. So what functions actually done by the cryptochrome? That is, first one, the two are generally involved in the regulation of the gene expression and obviously the circadian clock, or you can say the circadian rhythm. And second one, it also inhibits the scale elongation of the plant by the blue light. Okay, so second one, this is the, it inhibits the scale elongation. Okay, and third one is the, it helps in the stimulation of the leaf expansion. So generally three functions I say here, or just briefly say here, and these are another functions how controlled by the cryptochrome. I also try to tell this in another video elaborately how the cryptochrome actually control this all the functions. Okay, so these are not brief, obviously these are elaborate topic and elaborate description. So in this video I cannot explain about that, so I explain you in my another video about the functions, how the cryptochrome control this kind of functions in the plant's growth and development. Okay, so this is our topic, cryptochrome is ended here.